Okay, hello everyone. Welcome back. So this is the week 4 of the NPTEL course Immunology, and this course is coordinated by Professor SKG Sudip Kumar Ghosh and Professor Agneyo Ganguly from the Department of Bioscience and Biotechnology, IIT Kharagpur, and I am Beas Mukherjee, a research scholar uh, in the lab of Professor Day in the same department, Bioscience and Biotechnology, IIT Kharagpur. Okay, now in last class, we were talking about the generation of antibodies. So there is this huge diversity of antigens that are found in nature, and these antigens can enter into our body through pathogens, right? And there are only limited genes in our body. So how is it that from these genes, so many diverse combinations of antibodies are produced so that they can bind to the antigens with very high specificity and affinity, right? So in the last class, we have talked about these different modes of generating antibody diversity. For example, there is this VDJ recombination, there was junctional diversity, there was light chain and heavy chain genetic recombination. Okay, and then we have also talked about somatic hypermutation in the CDR regions. So what we have understood until now is that at the complementarity determining region of the antibody, majority of the mutations happen. Because of this, the CDR regions, especially CDR3, this is becoming very variable, and it is able to bind to different antigens of different protein sequence with very high affinity and specificity. Okay, in today's class, we are going to talk about something called isotype switching, which means that initially the antibody was produced in one particular uh, class, okay, and then from that it is forming different classes of these IG, A, IgD, etc. All of these different other classes are getting formed. Okay, we are going to talk about that, and we are going to talk about BCR and TCR, that is B cell receptor and T cell receptor, and we are going to talk about MHC in details, okay, major histocompatibility complex. So these are the few things that we are going to discuss in today's class. So for recap, we will start with the basic structure of antibody, and as you remember that the complementarity determining region was involved in the paratope region of the antibody, which was binding with the epitope of the antigen, and it was giving you this high specificity of the binding, high affinity of binding along with specificity. Okay, now this is a figure that explains this process. Now the antigen is shown in gray color and the epitope of the antigen is shown in black color, and the CDRs of the heavy chain, they are mentioned as CDRH, and CDR of light chain, they are mentioned as CDRL. Okay, and you can see that it is the CDR regions which is most variable, because this needs to come in contact with the epitope of different antigens, and the rest of the antibody may be having similar protein sequence. Okay, Now we are going to talk about class switching in details. Okay, so let's go back to the development of B cell, because here we will see exactly in which stage of the B cell development what process is happening in the antibody or the B cell receptor. Okay, so we will start with the hematopoietic stem cell, and then there was this lymphoid cell production. Now, all of this is within the bone marrow. Okay, you can see either one of the two diagrams, they explain the same thing. So in the bone marrow there is this gene rearrangement that is happening in the pro B cell stage, where heavy chain gene rearrangement is happening. And then in the pre-B cell stage, you can see that the light chain gene rearrangement is again happening. Okay, and in the immature B cell, what happens? RNA processing happens. So alternative splicing happens, and then mature B cell is getting formed. And then it comes in contact with the antigen, and in this stage, it is either IgM or it can produce IgD as well. So it is just two varieties, IgM or IgD. Okay, then what happens in the lifetime of this particular B cell, that all the other different classes of antibodies are produced. So we are going to look at that today. Okay. Now see what happens after antigen stimulation. This B cell gets activated and it undergoes differentiation. Okay. Then what happens? Then the class switching happens, and this happens in the peripheral lymphoid organs. So after class switching, you can see that IgG gets formed, IgA gets formed, IgE gets formed. All of these different variants get formed after class switching. Okay, and then of course plasma cell and memory cells will be formed. So we are going to talk about these variants now. Now let's see how they are formed and what happens. So one variable region, 5 possible isotypes. So antigen specificity is set by the variable region formed during VDJ recombination. So the specificity of the antigen, that is completely dependent on the, so FAB region is basically formed by the VDJ recombination that we have already discussed in our previous class. Okay. The heavy chain constant region gene, it has different variants. For example, C mu, C delta, C gamma, C epsilon and C alpha. These are all Greek letters, right? So these determine the isotype. So which gene is going to come to this VDJ, that tells you what kind of isotype will be formed for this particular antibody. Okay, and the constant region of this antibody, so this FC region, that is responsible for its effector functions. We know what are the effector functions: opsonization, neutralization and complement activation, right? So these are the three effector functions of an antibody. Now let's see that C mu, this actually forms your IgM, and what happens, this is the first responder. So this of course is a pentamer, and this is the strongest complement activator. Now different antibodies have different roles. Okay, so we will talk about that also in today's class. Then there is your C delta. So C delta gene, this forms IgD, and this is co-expressed with IgM on the naive B cell and B cell receptor as B cell receptor. Okay, then there is C gamma, which forms IgG. This is the most abundant in the serum, and this has the longest half-life, and most important property of IgG is that it crosses placenta, so it can come into the fetus from the mother. Okay, then there is C epsilon. Now this one forms IgE, which is responsible for allergic responses, and it is lowest in the serum. And coming to C alpha, this one forms IgA. So this is secreted in the form of a dimer. Okay, and this is the only isotype crossing epithelial barriers. So in our mucus secretions, in our gut, all of these places are where this particular antibody is secreted. Okay, now we will look at the gene. Okay, so we saw in last class that there was this locus in the chromosome. Locus means a specific location in the chromosome where all of these different genes were clustered together. So starting with the VDJ genes for heavy chain, and then there were the different constant region genes. Okay, So it's different in case of mouse and different in case of humans. So you can see that, okay. So after VD and J recombine, you have your C mu, C delta, then C gamma, 3, 2, so 1, 2, 3, and 4. There are 4 variants of the C gamma, and then C alpha also has 2 variants, okay. And you can see something very interesting there. There is another gene which is referred to as psi C epsilon. Now this is a pseudo gene. So what exactly is a pseudo gene? So a pseudo gene is a gene which does not actually give a functional protein. Why? There can be multiple reasons. For example, that gene can have a premature stop codon inside it. So what happens? Translation does not give a functional protein. Or this gene, so it may not be transcribed, okay, because it may lack a promoter sequence, okay. So we will look at this. Now genes are prefixed with psi. For example, psi C epsilon in humans are pseudogenes present but either not transcribed or not correctly translated, as it either lacks a promoter or it contains a premature stop codon. Okay, so this is the thing about human genome. There are a lot of things, but why they are present in that specific location, we still haven't figured out. Okay, and that is why biology is such an evolving field. Okay, now we will look at all of these 5 isotypes that you can see, okay. Now there are monomers, there are dimers, there are pentamers, and again these are joined by J chains. J chain is there to join all of these different antibody molecules together, okay. And we will look at these again in a little more details. So see, they are having their biologically active form, and IgM is a pentamer, IgG is a monomer, IgA is a dimer, and IgE is a monomer again, right? Now what is their molecular weight? You can see that all the different molecular weights are given. Coming to valency, I'm sure you remember that there were these FAB regions, and the FAB regions were able to bind to these antigens. So how many antigens it is able to bind, that gives it its valency. Okay, so if it's a dimer, it is going to bind to 2 antigens. If it is a pentamer, it is going to bind to 10 antigens, right? Coming to serum level. So which one is the lowest in the serum? We know that IgE is present in the lowest concentration in the serum. IgG is present in the highest concentration in the serum. Okay, half-life, if it is present in highest concentration, that means it will have the longest half-life, right? That is why it is still persistent in the serum. And IgE, once again, it is very less, and that is why it extinguishes, right? And then what happens? Complement activation. Now, for complement activation, complement system is again of different types, okay. One is called classical pathway, the other one is called alternative pathway, and the third one is called lectin mediated pathway. Now we will talk about complement system later on in another class. But these antibodies, they are very useful in activating the complement pathway. So IgM activates your classical pathway. IgG mainly it is involved in activation of alternate pathway, IgG1. Okay, if it's good if you can remember, it's okay. If you cannot, no problem. Okay, now we will go to crosses placenta. Remember this one is important, okay. So IgG is the one that crosses placenta. And key role, you can see that IgM is involved in primary response. So that means when the antigen enters into our body for the first time, that is the primary response. And at that time, IgM is most active. Then in the secondary response, IgG is most abundantly found, okay, and its effect is long term. Then IgA, it is secreted in the mucus and other secretory excretions in our body, okay. And IgE is important for allergy and antiparasitic response. Now IgD is very similar to IgM in most respects, but this is present at very low serum concentration, and it does not act as a secreted effector antibody. Okay, now we will move on, and you can see that in mouse, these are the different C constant region variants genes are given, and what happens is the primary transcript gets formed. Acha, so which one is going to come and bind to this VDJ? that is also directed by something, and we are going to talk about that later. Okay, so that is done by polyadenylation, and then RNA splicing happens. Okay, so we will talk about all of this in today's class. So you can see that this entire gene forms one primary transcript, and then there is alternative splicing. Okay, and after that there is translation gives your desired proteins. So here we can see that heavy chain and light chain are both getting formed. Okay, now we have a small question. Try to answer this question. A pseudogene such as psi C epsilon found within the human immunoglobulin heavy chain constant region locus is best defined as. Okay, you can answer in the chat box like we always do in this class. So A is a gene segment that has undergone somatic hypermutation to the point of, yeah, B. Okay, so DNA sequence with, okay, great. So all of you are answering. Very good. Let's see which is the right answer. Yes. So B is the right answer. Good. Okay. So it can either lack the promoter sequence, it can have this stop codon inside, okay, because of which it is never giving you a functional protein. Now, we will go to IgM and IgD. They share one single pre-mRNA. Okay, so what does that mean? So see, your C mu and C delta, they sit side by side in the genome immediately after this VDJ with a poly A signal right after. So after C mu, there is a poly A site. So poly A site means that there are multiple arginines. Sorry, not arginines, adenines. So multiple adenines are there, right? And then you have this C delta gene, and then once again multiple adenines. Now this poly A is useful. Why? Because poly A is the place where your transcription halts, right? So so transcription translation, they halt basically. So a single primary transcript is spliced two different ways to give two co-expressed receptor isotypes. So you can see this genomic DNA, this undergoes transcription, and then you have this pre-mRNA, and the pre-mRNA is having everything, right, all of these. And then what happens? It can be spliced in two different ways. So it can either be VDJ and C mu only comes, so that gives you IgM antibody after translation. Otherwise, this C mu portion can be skipped off, and it can C delta can come and join, and it is giving you IgD. Okay, now this is co-expressed with IgM on the surface of the mature naive B cells, and it is having again very low serum concentration, and it is not secreted. Now why this matters is both IgM and IgD are on the same B cell as BCR or B cell receptor, and they carry the identical variable region. So the VDJ is exactly the same. Okay, so both recognize the same antigen. Antigen binding to either together with a second signal from helper T cell triggers B cell proliferation and differentiation into the plasma cell. Okay, so we will look at this in a figure now so that you can understand this better. Now see, what happens is initially your pro B cell stage, what is happening? Initially there was J and D that are coming and recombining and giving rise to your pro B cell. Then V comes and joins with D. So it becomes VDJ. Okay. And this is found in the pre B cell. Then what happens? J and L in the immature B cell. So basically constant region comes and you get this IgM in the immature B cell. And in the mature B cell, what happens? IgD can also come and it can be expressed on the surface. So it is either IgM or IgD. That is why it is shown in two different colors. Okay. Now, so see, look at this. Now BCR, this B cell receptor can either be present as a membrane, it can be either present on the membrane, or it can be secreted out in the form of an antibody. So now what is the difference between these two? Everything is same in BCR and antibody, only there is only very slight difference. So we will look at that now. Okay, so you can see that there is this BDJ region, then your constant region starts. Okay, and we saw that there was this C mu and then there was this poly A site, right? So there is something called S exon. So what is exon? Exon is a gene segment that is going to get transcribed. Okay, introns are gene segments that are never transcribed into mRNA. So that is the difference between exons and introns. Okay. Now this S exon, S stands for secretary. So if this S exon and after that there is this poly A site once again, and poly A site can be named as PAS for our understanding. And then after that there are two exons again. There is this M1 and M2 exon. So M1 and M2 corresponds to membrane 1 and 2. This is basically this two code for the transmembrane region that is required for any antibody to be embedded in the membrane. Okay, now the difference between something that gets secreted and something that gets remained embedded in the membrane is definitely this transmembrane region. Now this M1 and M2 code for this transmembrane region plus a cytoplasmic tail. Okay, and the secretory portion, this codes for this signal sequence that is allowing this particular protein to be exported out and get secreted. Okay, now this is the secretory exon which is allowing it to get secreted, and this M1 and M2, this is allowing it to get embedded in the membrane. Okay, so polyadenylation happens at site 1 and site 2, and then RNA splicing happens, and it either makes this S part to come and join, and then you get this secretory antibody. So that means the antibody molecule is going to get secreted. Otherwise, mRNA encoding membrane, mu comes and mu chain. So this one M1 and M2 come and join, and you're going to get this BCR which is going to remain attached on the surface of this cell. Okay. Now, how do they look like? So in this figure, you can see that this is the entire amino acid sequence, and you can see that encoded by S exon. So this is the sequence that is basically encoded by the S exon, and this is for secretion. So you're seeing that there are both hydrophilic and hydrophobic amino acids. Now, if it is supposed to be secreted, that means it is going to come into contact with water, because in the cell there is this physiological pH, right? There are ions, and for the molecule to be soluble, it needs to have hydrophilic amino acids. So these are all charged amino acids that are going to interact with water molecules, ion molecules, and that is how it is going to be soluble. That is why it is having both hydrophilic and hydrophobic amino acids. But when you come to this membrane, M1 and M2, these two code for transmembrane region. Now transmembrane region is majorly having hydrophobic amino acids, because this is going to be embedded within the membrane, and there is this lipid bilayer, and it is of course hydrophobic in nature, right? And then there is the cytoplasmic tail which is more hydrophilic. So more hydrophilic amino acids are coming and sitting there. Okay, that is how your two different types of antibody molecules are formed. Then you have alternative splicing in a little more details. So see, what happens is receptor or antibody from the same gene it is getting created, but whether it is going to remain as a receptor or whether it is going to form an antibody, how is that determined? So the B cell receptor and the secreted antibody are having once again the same VDJ region and constant region transcript, but that constant region transcript gets spliced differently. So a membrane exon pair M1 M2 or a secretory tail, that is SC, is chosen depending upon the activation state. Okay. So initially in the resting or naive B cell, splicing includes this M1 and M2 exons, that is the transmembrane domain, and the SC secretory exon is skipped. So when it is a naive B cell, it hasn't met with the antigen yet. So there what happens, it is present in the form of this transmembrane antibody, right? So that is BCR, B cell receptor. The product is a transmembrane immunoglobulin anchored in the plasma membrane as B cell receptor, and the cytoplasmic tail signals upon antigen binding. and this stays on the cell surface as BCR. Coming to the next one, that is activated plasma cell. So splicing includes the secretory exon with its poly A signals M1 and M2 are skipped. Okay, so this M1 and M2 region is skipped and this S region is included after this VDG region. Then what happens? So VDG, then you have this C mu. It can be 1 to 4, any one. So 1, 2, 3 or 4, and then the SC comes and sits. The product is a secreted immunoglobulin, short hydrophilic tail, replaces the transmembrane domain. So the antibody is released into the blood. Okay. Now this is secreted into the blood as a free antibody. Okay. Now try to answer this question. So naive resting B cell simultaneously displays IgM and IgD as B cell receptors of identical antigen specificity. The molecular basis of this B. So alternative splicing of a, okay, let's see which is the right answer. Yeah, very good. So B is the right answer. Okay, so alternative splicing of a single primary transcript containing VDJ, C mu and C delta in tandem initially was there, and then after that you're getting the different variants in the plasma cell. Okay, we have another MCQ. So this one says, which of the following correctly explains why the resting state, that is the membrane bound and secreted forms of IgM, differ in structure despite arising from the same gene? Okay, so see the different options are, oh, see you guys are answering very fast, good. All right, so let's see which is the right answer. Yes, so once again B is the right answer. So basically alternative splicing is the one that selects between the two, right? Yeah, good, great. Now, this entire thing is explained once again. Okay, so you can see that this M1 and M2 come and join, and then what happens? Polyadenylation of the primary transcript happen, and you're getting this either mu M transcript or you're getting delta M transcript. Okay, and that is giving IgM or it is giving IgD. Now, we have 3 levels of antibody variation. Now, we were talking about antibody variation, right? So, we have talked about class switching. Now, besides class switching also, there are other variants of this antibody. So, what are the different variations of antibody? Now, this is a very important topic because since the terms are kind of confusing, this is one of the examiner's favorite questions. So, please try to understand this and remember this, all right? So there are 3 different levels of antibody variations again. They are isotypic, allotypic and idiotypic. Now what are these? So isotypic means that the constant region is different. The one that we talked about just now, right? So FAB region is the same, but your constant region is basically different. So two antibodies differ in the isotype. For example, IgG versus IgA. Variable region may be identical or different. Every individual has all isotypes. We all have all 5 isotypes of antibodies within us, right? Then you have allotypes. Now what is this allotype? It is the same isotype but different allele. Now what is allele? Allele means that what is the different kinds of gene population that can be present in a population. Okay, it is not inside one individual. So in one individual, there are only 2 sets of chromosomes, one from the mother and one from the father. But in a population, what are the different variants of a particular gene that are possible? So that makes up the alleles. Okay. Now, two people's IgG are both IgG, but the sequence differs slightly because they inherited different alleles of the same constant region gene from the parents. So that is allotypic variation. Then you have something called idiotypic variation. Now this one is very important. So see what happens in idiotypic variation is different variable regions. Okay. Now, two antibodies of the same isotype, but they are raised against different antigens, differ only in their variable region or hypervariable region rather. Then they are called idiotypes. So the constant regions are same, but the especially the hypervariable regions, these are slightly different because they are raised against different antigens. Now see what happens because of this. Now there is something called the anti-idiotypic response. Okay. Imagine this way that we know that whenever there is a patient who has been bitten by a snake, right, immediately this person is rushed to the hospital and they are given this anti venom. So this anti venom is given, right? So what is this anti venom? Anti venom means that a horse has been taken and the horse has been injected with this particular snake toxin, right, snake venom, and the horse's body produces these different uh, like antibodies against this particular venom, and that is taken, and that is your anti-venom, after all, anti-venom serum, and that is taken and that is given to the human. But then the snake venom that was given to the horse can be slightly different from the one of the snake that bit this patient, right? It can be slightly different. So then what is happening? Then the two antigens are actually different. Even though they are the same species of snake, there can be very slight difference. So these two antibodies, the one that the patient's body is producing because of the bite from the real snake, and the one that we got from the horse and that is being injected into the your patient. So both of these are very slightly different. And what happens because of this is, when a foreign antibody is injected, for example horse antivenom or let's say anti-tetanus serum, the recipient's immune system recognizes its variable region as foreign. So what happens is, this horse antivenom has been taken and the patient is also producing some kind of antibody against this real snake's real snake's venom. Then what happens, since this particular antibody was slightly different, the FAB region was slightly different, the patient's body saw it as an antigen and it is attacking this antibody. So what is happening? All this effort is now wasted and the patient is suffering from a different level of immune response, which where what is happening? This external antivenom is only getting uh, like destroyed by the patient's own antibodies. So this is why it is said here, see, that this is why repeated use of animal derived antisera loses effectiveness over time and is a key mechanism proposed in some autoimmune conditions where the antibody against a particular self antigen, for example anti-insulin antibody, it can itself condition where antibody against a self antigen, example anti-insulin antibody, can itself provoke an anti-idiotype reaction. So we will look at this in the next slide, okay, in a little more details. So see, these are the different variants, isotypic variants, where you see that the constant region is different between the two, allotypic variants where the alleles are different, and you have idiotypic determinants where the FAB region is different. So the one that is different is colored in black, okay. Now the one that we were talking about, this anti-idiotypic response. So see what is happening. What is an anti-idiotype antibody? An antibody raised against the idiotype, that is the variable region determinants of another antibody, is called an anti-idiotype antibody. Okay, so idiotype is basically an antibody's unique variable region. An anti-idiotype is a new antibody made against that variable region. So this horse immunoglobulin thing, I hope you understood that in anti-venom therapy, what happens is horse IgG is taken and this is our idiotype. Okay, and this neutralizes the snake venom very fast. But human anti-idiotype. So the human is now producing antibody against this horse antibody. And this targets the horse immunoglobulin itself. The effect fades on repeat, risk of reaction rises. Okay, coming to the second one. So this is an autoimmune insulin example. So see what is happening here. 1 second. Yes. So what happens here is, there is this anti-insulin antibody, which is an idiotype, and it is blocking immune signaling. Then what happens? Anti-idiotype therapy is done. So for example, inside your body, let's say there is some mutated protein. This mutated protein is acting as a, this is a basically an antibody which is preventing immune signaling. Okay. And so what happens? Uh, Therapeutically, a different antibody is made, and this is an anti-idiotype antibody, and this one goes and binds, and this one goes and basically prevents this other antibody to bind to this immune signaling proteins and block it, and therefore insulin is free to act now. But then what happens? Anti-idiotype, that is neutralizes the therapy. So again our body makes another antibody and it goes and neutralizes the therapy. So that is a very complicated thing that happens, and this is how the immune system is ever evolving, right? So it is something that acts very fast and something that undergoes mutations very fast, and what mutation can lead to what consequence, like it is very difficult to determine that, and that is why research in immunology is once again very evergreen. Right. So general principle is that any antibody's variable region can itself become a target. So idiotype, then what happens? You have this anti-idiotype and then anti-idiotype and so on. Right. Now we will go to the MCQs. So the MCQ says two immunoglobulin G antibodies isolated from two different healthy individuals show very minor amino acid sequence differences confined to polymorphic positions within their otherwise identical immunoglobulin constant region. Okay, unrelated to antigen specificity. This type of variation is called what? B. B is allotypic variation. Yes, good. So whenever you will see that there is something called polymorphism, it is because of allotypic variation. Okay. Acha. Now we have another one. So horse-derived antivenom is highly effective for a single acute um, snake attack, okay, but becomes progressively less effective and more prone to adverse reactions. B, okay. Let's see. Now what happens is if the same patient requires repeated antivenom treatment over time, this is best explained by. Good. So B is the right answer, right? Correct. Now we'll go to the next one. So IgM circulates predominantly as a pentamer with antigen binding valency of 10. This we know, avidity is very high. Therefore, now while serum IgG is a monomer with a valency 2, which statement correctly relates the structural difference to IgM's role in immunity? Okay, so we have varied answers here. Let us see which is the right answer. Yeah, so the right answer is A. So A is the right answer here. Okay, the figure that is given, it is not in correlation with this diagram. So I am going to remove this figure. This figure will come for your next question. Okay, it is not for this particular question. Okay, so see, higher valency compensates for typically lower binding affinity per site, making IgM an efficient early. Oh, acha, agglutinator is here. Okay. So what is agglutination? We will actually look at that. So I think it was, acha, no, it is there for this question only. Okay. So this makes it a good agglutination. So agglutinator. So what is agglutination? We will look at that now. Okay. So basically what is agglutination? It means that antibodies, they come and they bind to these antigen and they make a clump. Okay, so you can see that basically agglutination is clumping of particles like cells or bacteria caused by antibodies cross-linking on the surface antigen. So they just come and more of them come and they cross-link together to make this huge complex, and then what happens? The whole thing becomes, the whole thing precipitates out. Okay, and that is called agglutination. So you can see that agglutination happens in blood also. and we will talk about this in details in the upcoming classes. Okay, there is something called hemagglutinin and all of these different proteins that are involved, and you can see that if you guys have done these experiments where you take human blood and then there is like antigen in the blood, right? And then you add some kind of antigen or antibody from outside, then so we will talk about this in the upcoming classes. So there you can see this agglutination of blood, and the figure is also given there. Okay, now we will go to the next one. Now we'll look at isotype switching once again. So unlike IgM and IgD alternative splicing. So for IgM and IgD, there is this alternative splicing that is happening. Okay, but what about IgA, IgE and IgG? What about these, right? So there is another level of alternative splicing. So these require physically deleting DNA. So happens in secondary immune response in response to what kind of cytokine environment is there. Depending upon that, the different antibodies are basically produced, or different immunoglobulins are basically produced. Okay, so repetitive DNA sequences called switch regions upstream of each constant region except for IgD recombine, looping out and eliminating everything in between. Okay, you can see how it is there on the DNA. So there is this VDG region, then you have this S mu, C mu, S gamma. So these S is basically the switch region. This is repetitive DNA sequences, right? So switch regions are there, and the switch regions S mu, S gamma, they recombine, and C mu basically loops out and is excised from the chromosome. So then what happened? The others come and join. So after switching, you have VDJ, then S gamma and C gamma comes and joins. So it becomes this uh, gamma comes and joins. So it becomes IgG. Right, so C mu is now permanently deleted. So if C mu would have come, it would have made IgM, but since it got deleted, then it can never revert to making IgM again, and S gamma and C gamma can later recombine further with S alpha converting IgG to IgA if mucosal secretory response is needed. Okay, now why this happens? Why switch isotype? That is because different antibodies are required for different purposes. For example, placental transfer. When that is needed, IgM is the only antibody that can do it. Then you have mucosal protection. So IgA is the only antibody that can cross epithelial barriers into the gut and the airway lumen. So the entire like the windpipe and everything, the place from which this goes. Okay, and then you have matched effector function. So IgM is the best when it comes to activation of classical complement system, and later responses favor the isotype suited to the site of infection. Okay, and then this is driven by cytokines. So cytokine signals control which S regions or switch regions will open for recombination, same recombination machinery as VDJ joining. So this entire process, somebody unmuted yourself and unmuted yourself. Okay, good. So let us continue. Now you can see that how is this happening? How are these S regions coming and joining? Once again, this is for mouse, okay? So these two are coming together and the rest of the DNA in between, this is forming a loop and then it is getting excised out, okay? And through a similar mechanism as recombination that we studied in our previous class. So this is how all the rest of it is going, and you can see here that C epsilon and C alpha are coming and joining. Okay, and that is how you can get the different isotype classes. Now we have a MCQ here. So a B cell has undergone class switch recombination such that its BDJ exon is now currently directly joined to C alpha with all intervening constant region deleted. Okay, which of the following isotype can this B cell alone and its progeny no longer produce even after further activation? So it is joined to C alpha, then what can it not produce? Okay, good. Let's see which is the right answer. Yes, good. So C is the right answer. Now it can produce IgA1 and IgA2. Okay, but then it cannot produce IgM, D and IgG antibodies. Okay. Acha. Now we have this MHC structure. Okay, so now we have talked enough about antibodies. We will talk about T cell receptor in a while. And right now we are going to start with MHC. Okay. Now MHC molecules, you know that they are major histocompatibility complex, and both classes of this MHC form a peptide binding cleft. Now we understand that MHC class 1 and class 2 are two different proteins. They present antigen on their surface, and your cytotoxic T cell comes and recognizes the peptides presented by MHC class 1, and the helper T cell come and recognize the peptides presented by MHC class 2. But how do they present the peptides? How do they bind to the peptides? And how do these other T cells come and recognize them? So we are going to talk about that in today's class. Okay. So see, your MHC class 1, it is having this, one second, yeah. So both classes form a peptide binding cleft from 2 alpha helices over an 8 antiparallel beta strand floor. Okay. They are assembled from different chains and changes how the peptide, how much peptide they can hold, which changes how much peptide they can hold. Now coming to MHC class 1, so there is one chain which is formed from alpha 1, alpha 2 and alpha 3, and there is another chain called beta 2 microglobulin, which is non-covalently bound to this alpha chains, and there is one transmembrane domain, obviously, because it is going to sit on the membrane, right? So the peptide binding cleft formed by, this is formed by a single peptide polypeptide, that is alpha 1 and alpha 2, and the groove is closed at both the ends. So this is how it looks like. Okay, the groove is closed by both the ends and it is formed by a single polypeptide. And because the groove is so small and so narrow, only peptides of 8 to 10 amino acids can be presented on it. Okay, it is coming from inside the cell and only amino acids 8 to 10 peptide can be presented on it. And how do these peptides bind? So the peptide anchors mainly at its two ends at hydrophobic residue. So the interaction between the peptide and this MHC molecule is through hydrophobic interactions, and this end of the peptide remains bound and middle is less constrained. Okay. Then what happens? This presents endogenous or intracellular antigens, for example, viral or tumor protein. This you remember. And this is recognized by CD8 plus cytotoxic T cells. Okay. Coming to MHC class 2, remember it this way, that there are two chains. Okay, one is alpha chain, one is beta chain. Both are holding it like this. So this is having a bigger groove. Okay, so two chains alpha and beta with two domains each. Each chain has its own transmembrane domain. The peptide binding cleft formed by the two chains are alpha 1 and beta 1, and the groove is open at both ends. And one second. Okay. So the groove is open at both ends and binds larger and more variable peptides. Okay. Now this is binding peptides of amino acids 13 to 20. Now the peptide contacts the groove along its whole length, not just the ends. And it binds more variety of peptides than MHC class 1. And this one presents exogenous antigens taken up by phagocytes or endocytes. Okay. And this is recognized by the helper T cell with CD4 receptor. Now, the peptide binding actually stabilizes the MHC. That is why in nature it is very difficult to get MHC that is free, unbound to any peptide, okay, under physiological conditions. So physiological conditions means that normal pH that is maintained in the body, your normal salt concentration of 150 millimolar that is maintained in the body, etc. That is called physiological condition. Okay. Now this figure is going to tell you how exactly MHC class 1 and class 2 look like. Okay, so you can see that this is MHC class 1, the other one is MHC class 2. This one has one transmembrane domain, this one has two transmembrane domains. Okay, and you can see that the cleft is very narrow and small for this class 1, but it is like longer for class 2, and so wider variety of peptides can be held by this, and also longer peptides can be held by class 2. So how they are formed, that is shown here. So you are seeing that they are having this transmembrane domain which is coming from TM, and they are also having this CC. So C is basically your cytoplasmic tail. So the C genes, they code for the cytoplasmic tail. Okay. Achha. Now, how does this MHC class 1 and class 2 present antigen? That is also given here. Okay, so you can go through this. One of them is definitely endogenous pathway, the other one is exogenous pathway. You can go through this if you're interested. Now we come to the MCQ. Okay, try to answer this one. Which statement correctly distinguishes the peptide binding groove architecture of MHC class 1 from that of MHC class 2? MHC class 1's groove is contributed by single peptide polypeptide chain and is closed at both ends. Bound peptide length MHC class 2 is contributed by two separate chain and it is open at both ends, allowing longer peptides. A. Okay, let's see which is the right answer. Yes, good. So A is the right answer. Okay, so that means you guys have understood. Great. Now we have another MCQ. So in peptide elution or binding motif studies of MHC class 1 molecules, bound peptides of otherwise varying sequences were found to consistently share a hydrophobic residue at the C terminus and another conserved residue at an internal position. Right, so this peptide was having this hydrophobic residue at the C terminus and another internal residue, while the other positions varied freely. This observation is best explained by. So you are saying that it is B. So MHC class 1 engages peptides mainly through small number of defined and anchored residues at fixed pockets tolerating variability. Okay, let's see. Let's see which is the right answer. Yes. So B is the right answer. That is because while we were talking about this, I forgot to tell you that in this cleft of MHC class 1, it is the major interaction is hydrophobic interaction by C terminal of this peptide, and there is a central interaction again with a hydrophobic residue. Okay, but for your MHC class 2, there can be not necessarily conserved hydrophobic interactions, but it can be any type of non-covalent interaction. For example, hydrogen bond, there can be van der Waals interaction, etc. along the entire length of the peptide. Okay, so that is why this is B is the right answer. Now, why are MHC so polymorphic and multigenic? So what is meant by polymorphic? Polymorphic basically means that they are very different across the population. Okay, so why is that so? Now see, the peptide binding cleft is far less specific than a T cell receptor or antibody, and one MHC molecule can display many peptides as long as a few anchor positions match a loose consensus pattern. Right. Now, broader peptide coverage. Of course, now MHC molecules, there are only two types of MHC molecules. They will have to present all different types of antigens, right? They cannot be so specific as your BCR or antibodies. Now, broader peptide coverage. So, multiple MHC gene copies per individual, each with different anchor residue preferences, together present a much wider range of processed peptides than a single molecule could. Okay, that is why there are multiple genes. Okay, now you have resilience to mutation. If only one MHC gene existed, a single mutation could collapse the entire antigen presentation machinery inside our body. And with it, the T cell activation would also go downhill. And then what happens? That is why multiple copies of MHC genes buffer against this. Okay, resilience to viral interference. So some viruses evolve ways to block these MHC functions. Now with several MHC gene copies, blocking one still leaves the others able to present antigen. Population level diversity or polymorphism. So MHC genes are among the most polymorphic in the genome across a population. Many different alleles. Allelic variants exist. No single pathogen can evade presentation in everyone. Okay. Now, how are B cell epitopes and T cell epitopes different? So B cell epitopes, uh, they are recognized. So epitope means that there is this antigen. Antigen is having this tiny portion with which the antibody interacts. Now for B cell and T cell, the epitopes can be slightly different. How is that possible? Even though it is the same antigen. Remember B cells, BCR, they are able to recognize the entire antigen, right? But TCR is only able to recognize the antigen once it has been presented by MHC. This is one of the major differences. Now see, the same antigen but two different rules of recognition. So B cell epitopes, they are recognized by the BCR or antibody directly. So engaged on the antigen's native folded conformation and must be accessible on the surface of the intact protein. No processing step is required and the recognition happens on the outside. But coming to the T cell epitope, this is recognized by TCR via MHC. So antigen is proteolytically processed into shorter peptide fragments. Fragments come from the region buried inside the native protein. So now these fragments, they are not necessarily present on the surface of this antigen, but they can come from any part of this antigenic protein, right? And they will be recognized by the TCR. So only the peptide loaded onto the MHC and displayed on the cell surface counts. So overlap is possible, but it is not guaranteed. So that means that a surface antigen can serve as both epitope for B cell as well as T cell receptors, but can only happen so this is kind of a rare phenomenon. The two recognition modes place fundamentally different structural demands on the epitope. So that even though the let's say that the surface residues get mutated in a virus, our body cells are still able to recognize it because this is after all going to get like cut off, and then all the buried residues and all the other residues, they are also going to come into contact with the T cell receptors, and that is how it is going to recognize that this is a foreign antigen. Okay, so that is why there are multiple levels of checkpoints. These are all of the different ways in which our immune system is protecting us every single second as we speak today, right? And that is why we should be grateful. Like, it is amusing, very amusing. The more you know, the more you realize how amusing our body really is. Now you have this BCR and TCR, and it is given in the form of diagrams, okay, for your understanding. And you can see that there are so many disulfide linkages, okay, so that now disulfide linkages are very strong interactions, so that the proteins do not get denatured very easily, so that the protein is not losing its 3D shape and biologically active form and geometry. It is held together by so many different interactions, okay. Now you have this TCR MHC pairing rule. Now crystal structures of this peptide MHC TCR complex, MHC antigen and TCR complex show an consistent orientation where the TCR sits centered over the peptide, contacting both the MHC helices with its conserved framework region. That is why framework regions are so important. If you remember that the framework regions had very low variability and the CDRs had very high variability. Okay, now MHC class 1 plus TCR. So there is this alpha 1 domain of MHC interacts with the beta region of the variable chain of TCR, and again alpha 2 domain of MHC1 interacts with the alpha variable region of TCR, and that plus there is of course the co-receptor. So alpha 3 domain of this MHC binds with CD8 co-receptor and restricts recognition to CD8 cytotoxic T cells only. Coming to MHC class 2, alpha 1 domain interacts with beta variable region, and beta 1 domain interacts with alpha variable region of the TCR, and the beta 2 domain binds to the CD4 receptor co-receptor and restricts recognition to CD4 helper T cells only. Okay, now role of the co-receptor, you can see that co-receptors are also very important in binding. Now the CD8 binds specifically to MHC1, CD4 binds specifically to MHC2. If this cross matching failed, example CD8 engaging with MHC2, cytotoxic T cells would misfire against cells that are not actually virus infected or tumorous, since cytotoxic recognition directly triggers killing of the antigen presenting cells, right? So this is also another level of another kind of checkpoint, right, inside the cell. And that is why all of these mechanisms are there to make sure that the immune system is not making any mistake, because even the smallest of mistakes can be fatal for the organism, right? Now you can see that all the TCR and its co-receptors are given. This is how all of these look like. Okay, you can have a look whenever you get time. Now you have this TCR formation. So how is TCR formed? Once again, there is VDJ recombination, constant region comes and binds, and two different chains, alpha 1 and alpha 2, are getting formed. Okay, that is how your TCR formation happens. Now we will try to solve this MCQ. Okay. Try to solve this one. Acha. And give me 1 second. Okay. Give me 1 second. Try to solve this MCQ. I'll be back. Okay, so we have answers. Great. So we have answers now. Right, so you guys are saying that this is B. So I'll read the question once again. So crystal structures of TCR peptide MHC complex reveal a consistent pattern of contacts, and for MHC class 1, which statement correctly describes this pattern? So you're saying that MHC alpha 1 contacts your B chain variable region of TCR, and alpha 2 contacts the alpha chain variable region. Let's see. Okay. Now you can check. This should possibly be. So basically we saw that it was alpha beta interaction and again beta alpha interaction, right? So you can check if this answer is correct. Okay. You can actually check this. So we can go back and we can check that. Oh yes. So alpha 2 is contacting your alpha of variable TCR. So yes. So this is correct. All right. Great. Yeah. So this is correct. Great. So we will move on to the next question. Right. So next, of course, there is this antigen presentation. Now there are two ways of antigen presentation. Of course, there is endogenous pathway, there is exogenous pathway. Endogenous pathway is taken up by the MHC class 1, and the protein is synthesized inside the cell. It can be a viral protein, it can be self antigen, and it can be a tumor cell. So again, self antigen of this tumor cell, and this is degraded in the cytosol and it is loaded onto this MHC class 1, and it is displayed virtually on all nucleated cells. So remember this, that all cells with nucleus will have MHC class 1 on their surface. Okay, now we will come to a very interesting story in some time. Okay, just remember that every cell that has a nucleus will be having this MHC class 1 on its surface. Okay, now what happens if a particular cell is not having MHC class, it is not having a nucleus. So that is the story that we are going to talk about. Okay, now see, this is recognized by CD8 cytotoxic T cells, and outcome is infected or tumor cell is definitely killed. Okay, now you have this exogenous pathway where we have this MHC class 2 molecules, and the extracellular protein is taken up by phagocytosis or endocytosis, and this is degraded in the endosomal vesicles and loaded onto the MHC class 2 molecule, and then this is restricted to professional antigen presenting cells recognized by helper T cells, and the outcome is that B cell activation is started. Okay, now antigen presenting cells such as dendritic cells, macrophages, B cells, the only three types of cells which constitute MHC class 2 expression, since they help to present these antigens to the helper T cells. Okay, so only specialized antigen presenting cells are having MHC class 2 receptors on the surface, and all other cells are having MHC class 1. They also have MHC class 1. Okay, now this is something very interesting. So you can see that these T cell receptors, they do not have very high, very strong binding with the antigens, right? Antibodies, they have a very wide range of binding with the antigens. It is not very strong. Okay, now that is why you can see that for TH cell and for TC cells, beside the T cell receptor, there are multiple other interactors that are involved in this interaction. So you can see that there is a CD2 and LFA interaction. So what is LFA? We will look at that in the next slide. Okay, and then there are multiple interactions. So there is a CD3 interactions, co-receptor interaction, there are CD28 interactions. So all of these are there. And the antigen presenting cell is once again having B7, it is having CD22. So all of these different interactions are going on. Similarly, for T cell also, there are multiple other interactions by the co-receptors and other molecules on the surface besides the TCR alone. Okay, so what are these interactions? You can actually read about that. So everything is given here in a lot of details. So if you want, you can go through these. Okay, now we will move on to this interesting story that I was talking about. So basically what happens is, where is MHC expressed and how one parasite hides. So you can see that in T cells, the level of MHC1 expression is very high, and in basically B cells, T cells, macrophage, dendritic cell, you have this MHC class 1 very high expression. Class 2 is low in T cells, and it is only there in activated T cells. B cell and macrophage dendritic cell, once again these are antigen presenting cells, so the MHC class 2 expression is very high. Then you have epithelial cell where class 2 expression is once again high, and other normal nucleated cells. So any cell that has a nucleus, there will be MHC class 1 even though there is no MHC class 2. But in red blood cells, MHC class 1 is absent. Why? Because the red blood cells, mature red blood cells, they lack nucleus. Okay, because they are carrying oxygen, so that they don't use up this oxygen, and to make sure that it is getting maximum it can travel very fast, and it can like travel through very narrow spaces and all. Because of that, it is having very special characteristics. RBC have very special characteristics. One of them is lacking a nucleus, and it performs no protein synthesis. So no MHC class 1 is required, and that is why it lacks an MHC class 1. Plus, they cannot phagocytosize. They do not have half of the cellular machinery also. Okay, and that is why they do not phagocytosize. So they do not have MHC class 2. So it does not have any MHC on its surface. Now there is this malarial parasite, Plasmodium falciparum. This one grows inside red blood cell. Now parasites, they are also very, like, strangely, they have adapted in such a way that these are actually parasites, right? So if the human body is evolving, the parasites will also undergo mutation, and they will also evolve simultaneously. So you can see that these actually grow inside the red blood cells, and the one cell type that is expressing neither MHC1 and no MHC2. So the same thing that is given here, it is given in the next slide also. So we can look at that, and then you will be able to relate. Okay, we have this nice figure here. So you can see that when the mosquito bites, mosquito is carrying this Plasmodium falciparum organism. Okay, it bites, and then it releases sporozoites. These are spores, and these come into the liver, and then what happened? They enter into the RBC in the form of merozoites, and these inside the RBC, they are like multiplying. Okay, so the result is that no infected cell antigen ever reaches the surface, and so cytotoxic T cells cannot detect or kill the parasite while it replicates inside the RBC happily. Then what happens? The only vulnerable moment is this actually ruptures your RBC. Okay, it ruptures the RBC, and it is kind of a cycle. So if you know anybody who has suffered from malaria, you'll know that fever comes at regular intervals, and at that time the person is completely non-functional. They are not able to do anything, and it is very terrible. Very high fever, chills are there in the body, and that is when RBC basically bursts open and the parasites are coming out into the bloodstream. It is only in that stage that the immune cells have a chance to recognize these antigens, antigens before they are once again entering into another RBC. So if they enter into another RBC, once again they will go unnoticed until the cycle repeats and the RBC is ruptured. So briefly, when RBC bursts and the parasites are extracellular, coinciding with the classical cyclical fever and chills of malaria, they may be identified by the immune system. Now, another very interesting thing about this parasite is that a large family of around 200 genes are there, of which only one expresses at a time of the variant surface proteins. Okay, this lets the parasite switch identity, undermining antibody-based vaccines. So even when it is coming out, and even when we are giving vaccines from outside, because there are so many different genes that are possible, and they can give rise to different antigenic variants, surface antigens, the vaccines are also not very potent, and the immune cells are also not able to recognize, right? And that is why, okay, now we have an MCQ here. So try to solve this one. So CD8 binds specifically to MHC class 1 via its alpha 2 alpha 3 domains, while CD4 binds specifically to MHC class 2 via its beta 2 domain B. Okay, which is the most important functional consequence of the strict pairing. Good. So B is the consequence, right? So you guys have understood. So if it is endogenous antigen, it is MHC class 1 and cytotoxic T cells recognize. If it is so if it is exogenous antigen, then it is MHC class 2 presented and the helper T cells recognize. Okay, great. Now we have another MCQ. Which set of cell types constitute the professional antigen presenting cells that constitutively express high levels of MHC class 2? Good. Right. So once again, B cells, macrophages and dendritic cells. The figure is given and you can see that B cells, they are presenting both MHC class 1, class 2, dendritic cells also both class 1, class 2, and macrophage also both class 1, class 2. So remember, it is not that they are presenting only the MHC class 2 cells, but they are obviously also cells with nucleus. So they are presenting MHC class 1 as well. Okay, they are making proteins, right? That is why they are presenting both the type of MHC molecules. Okay. Now, coming to the last few questions. Why do virtually all nucleated cells in the body express MHC class 1, whereas MHC class 2 expression is restricted to only a small subset of specialized cells? A. Okay. MHC class 1 is needed to present any endogenously synthesized, including viral or tumor proteins for cytotoxic T cell surveillance, is essentially any cell type, whereas MHC class 2 presentation of phagocytosed in extracellular antigen to helper T cell is a specialized function required only for professional antigen presenting cells. Great. So you have understood that that A is the right answer. Now give me a second and do not see the answer. Oh, this I think was your last. Okay, 1 second. Now you have another question and let's hope you did not see the right answer. Okay, try to answer this one. During the intra-erythrocytic stage of its life cycle, acha, I hope you have read and then you're answering. Okay, so during the intra-erythrocytic stage of its life cycle, Plasmodium falciparum evades the T cell mediated immune surveillance chiefly because, yes, so mature red blood cell lack nucleus and do not express MHC class 1 or class 2. So the parasite antigen produced during this intracellular phase cannot be displayed on the cell surface for T cell recognition. Okay, so with that we have come to the end of today's class. And that is the right answer, correct. Now these are the references once again, okay, and I have added the references for today's class also, plus you have all of these MCQs that were there in the previous PPT as well, and this will be shared with you. Okay, so if you have any questions, you can ask. Otherwise, we can end today's class. Thank you so much for listening all this while. Thank you so much for answering all the questions. I'm very, very flattered that you guys are actually listening and answering the questions so promptly. Very good. And I hope I am able to teach you the things that I have understood until now. So we will meet in the next class. Okay. And yeah, thank you. Thank you so much. Thanks to everyone. So we can end the call if nobody has any more answers. And I've already given you my mail ID. So if there are any doubts, any issues, you can mail me regarding anything. Okay, any feedback, anything that you want. Okay, and then I'll definitely answer. Okay, thank you so much. I'll see you again for the next class. Bye-bye.